Hello and welcome to Biousage Lessons. Last class we were talking about genes which are going to be happening in DNA, which are going to be making chromosomes. So today we're going to be talking about genes and chromosomes. So first, prokaryotes. Prokaryotic chromosomes, they are not like our chromosomes in eukaryotes. They are going to be different. If you remember, the structure of cells in prokaryotes, they are going to be having lots of these structures, cell membrane, cell wall, this slime capsule, and there in the nucleoid, in a condensation of genetic material, you're going to find the DNA, which is going to be circular. So this is something that's different in prokaryotes than in eukaryotes. The prokaryotes have circular DNA, and this DNA is going to be naked. Naked can be because it's not surrounded by a nucleus, but mainly it's not coiled around proteins. If you get the DNA, you're going to see only DNA in prokaryotes, not proteins like we see here in eukaryotes. You can also find tiny little chromosomes sometimes when these prokaryotes, they are exchanging genetic material, which are called plasmids. So this here is the structure of prokaryotic DNA. Okay, so naked, that's no nucleus, and also no histones rolling around it, so no proteins, it's only pure DNA. There was an investigation called the Carnes investigation, or Carnes technique, that they did an autoradiography. Basically, in this technique, the scientist, he made the bacteria to grow in radioactive nitrogen medium. So the DNA was built radioactive. The bacteria that was used there was E. coli, a prokaryote. Okay, so you have there radioactive isotope of hydrogen, actually not nitrogen. He broke down the cells, so lysed the cells, and he covered the slide with films. And they kept there in the dark for 2 months. What did he see? That image you are seeing on the left. It's roundish, but not absolutely roundish. There's something happening over there. We can see here that there is one thing here, apparently here as well, but here you have something thicker going on in there. So what actually is that? If you see several pictures, you see several stages of this, but the interpretation of that is going to be this. The first stage of replication is going to see that you're going to have DNA that will be half radioactive, half not. So you're going to have that thin line over there. But then during the process of replication, you have this labeled DNA, the radioactive one, that's being copied over here, and it's thicker because now you have a double helix over there. This radiography shows that the DNA is circular. It also shows that the DNA is being copied, and it is copied only from one point on all the way through. It's different from eukaryotes, which chromosomes are copied, starting several different positions at the same time. So you have multiple points of origin of replication in eukaryotes. In prokaryotes, only one. It starts in one tip, goes all the way around until you get to the other end. This is the Carnes autoradiography showing this prokaryotes ribosome. In eukaryotes, like us, what you're going to be having is that the DNA is coiled around histones. This stuff here that's about 10 nanometers wide is coiled around there, and then it coils more and coils more until it makes this image here that we know as a chromosome. Chromosomes, in certain part of the stage of the life cycle, they look like this axis. Okay, and they are still small. They have about 7 nanometers in diameter when we are capable of seeing them. Here, to show that the DNA is wrapped around these histones, let's take a look even closer. There in the white we have DNA without histones, and in the black we have the histones with DNA coiled around them. These histones, they are responsible for making the chromosome when the chromosome is coiled, when the chromosome is in this X format, ready for cell division. But also, these histones, they help in the expression of genes. If the DNA is coiled around the stones, it cannot be transcribed, therefore cannot be translated. If it's loose, like in the white arrow over there, then it can be transcribed, and therefore it can be translated, producing proteins. So histones have two main tasks. One, regulating the expression of the gene, and also it helps to condensate the chromosomes for cell division. Okay, so we are talking about genes and now we're talking about chromosomes. So we are going to be having here a table I want to show you with organisms, the number of protein coding genes. So not genes that do other stuff like genes that make transport RNA or genes that make ribosomes, genes that code for proteins, enzymes and other stuff, okay? And the size of the genome in million bases and the number of chromosomes that we have there. This is the diploid number, the number that you find in the typical regular cell. And bacteria, they are very, very primitive and they also don't have many genes. So Escherichia coli, the bacteria that was investigated using the Karen method, has about 4,000 protein coding genes. It's not little, 4,000 is quite a lot, okay? And it has 5 million bases, 5 millions to code for 4,000 genes. And the number of chromosomes, of course, is 1 because it's circular. There's no diploid number here because it's a bacteria, okay? Discounting, of course, eventual plasmids that can be found. Humans, okay, this is us. We have about 20,000 protein coding genes. It depends on what you call protein, depends on what you call gene. Some authors are going to say that there's more, 30, 40,000, but this is a number that most people, they orbit around, they gravitate around this number. 20,000 genes is something widely accepted for humans. And how many million bases do we have? 3,200 million bases. So basically we have about 3 billion bases in our genome. And that's located in 46 chromosomes. Ooh, 46 tapes of DNA. What about other organisms? Rice. Oh, rice is not as complex as we are they? So they should have less genes. Ooh, no, I'm sorry. They have more genes. They have 35,000 genes and they have a smaller genome. Also, they have less bases, but they have more genes. That's quite weird. And what about the number of chromosomes? They have less chromosomes, 24. They have 11 pairs of chromosomes. Okay, wheat is quite similar to rice, so it should be the same stuff over there. Oh no, 100,000 genes. Oh man, this stuff is really full of genes. And it's also full of bases, 16 billion bases in the wheat. And all that in 42 chromosomes. Yeast, very simple fungus, unicellular, very simple stuff. So you should expect that things should be simple here. And they quite are, 6,000 genes, so much less than humans. And that located in not many million bases, so 12 million bases. So yeah, quite a few, but also have 32 chromosomes. Okay, wild mustard, a plant. Okay, another plant, and about the same number, a little bit more than we expect to find in humans, number of genes, but a smaller genome and 18 chromosomes. Okay, mouse, okay, we are coming back here to mammals. Mouse is a mammal just like us, so should have, oh look, a little bit more genes than we have, and a little bit less bases in the genome. And how many chromosomes? A little bit less chromosomes than we do have. Whoa, what about dogs? The best friend of us humans, unless you're a cat person. About same number of genes that we have, about the same number of bases that we have in a mouse close to humans, but they have many more chromosomes. I don't know if you realize, but there is not a very clear pattern here. Of course, bacteria are very simple. They have less genes. So you can say that very simple genes, the simple organisms have less genes than more complex organisms. But after that, things become quite a mess. Okay, you have rice, wheat with many genes, and humans and dogs and mouse, mammals, very complex stuff with less genes than they have. So there's not much of a clear pattern. And also, there is absolutely no pattern here, apart from bacteria having a circular DNA. No pattern here for the number of chromosomes that we find in eukaryotes. So, not very clear. Although the size of the genome kind of correlates positively, with some exceptions. Of course, there are many exceptions here in eukaryotes, and here it's a little bit wide, so not a perfect correlation, but there's quite a positive correlation between the size of the genome, the number of bases that are there, and the number of protein coding genes. So the bigger the genome, apparently more protein coding genes are going to be present in there. Okay, this is quite expected because more genes, more DNA, more genes could be expressed there, but it's not an absolutely rule over there. Okay. Now let's talk about the chromosome. This is going to be very important for us to understand cell division, mitosis, meiosis, and also to understand the genetics. Those images here on the left here and on the right here, these here are representations of chromosomes in different stages of the cell life. What we have here is that on the left we have the chromosomes before replication, and on the right we have chromosomes after replication. So actually what you see here, this and then this, actually is a perfect copy, or should be perfect if there are no mutations, of this one over there. And what you see here, this over there and this over here, is a perfect copy of what you have over there. So basically these are the same thing. These are chromosomes, okay? And in yellow over there, I'm representing the place where the two sides, after being copied, they are glued together. It's the center of the X in the chromosomes when they are replicated. This is called the centromere. In Portuguese, it's going to be centrômero, but yeah, centromere. Okay. Those two guys over there on the left, they are what you call homologous chromosomes. They are chromosomes of similar size, similar band pattern, similar. They should be the same. They are not exactly the same because we receive one from mother, one from father, and actually mother and father, they have some slight variation around the genes in the chromosomes. But so these are the chromosomes. This is what we're going to be calling a pair. So homologous chromosomes, and after replication, they are also homologous chromosomes. The chromosomes on the left, they are before replication. They have only one line over there, and on the right, they are forming this axis. So let's take a look on what these lines are. So what we have here, each of these lines over there, so this stuff here and this stuff over there, these are called chromatids, which is the supercoiled tape of DNA, which after replication, we have two chromatids in the chromosome. So we have sister chromatids. This should be absolutely identical unless there was a mutation in the moment of the copying. And before we had here, these are also chromatids. So before replication, each chromosome is made by one chromatid. After replication, each chromosome is made by two chromatids. So this is it. Chromatid, the line that you see over there, one before replication, two, or making an X like this here, after replication. Okay, so this here is another image, and we have here in blue the one that came from your father, this here the one that came from your mother, this same here, this here came from your father, this here came from your mother. This on the left is before replication, on the right you have after replication, and you need to learn these names, okay? So chromosomes and chromosomes, these are homologous because they are quite similar. When I show you the karyotyping, this will be a little bit more clear. And we have one chromatid before replication in each chromosome, and you have two chromatids in each chromosome after replication, okay? The table here again, just to stress out that the number of chromosomes, they mean absolutely nothing. You can have a lot of small chromosomes, you can have few larger chromosomes. Chromosomes are just the way that the tapes are organized. They're not going to have much influence on our life. It has nothing to do with complexity, okay? Another example here, or a few more for you. Homo sapiens, our close relative, chimpanzees, 48 chromosomes. So we have different number of chromosomes in different organisms, and without correlation with complexity or whatever, okay? So, as I talk about chromosome, we talk about karyotyping. Karyotyping is a technique to visualize chromosomes. Basically, we can take pictures and we see, like here in Zea mays, which is corn, we can see that there are 10 pairs of chromosomes. So Zea mays have 20 chromosomes. While Canis familiaris, like I showed before, they have 38 pairs of chromosomes. So they have actually 76 chromosomes. Okay, so we can see the chromosomes using this technique of karyotyping. And as you can see here, we have chromosomes coming in pairs. A pair, another pair here. You see that the chromosomes may have different sizes. Like here, we have kind of a long one in dog there, very small ones over here in the end and over there. And karyotypes, they are organized, chromosomes are organized in pairs, and they are ordered from the biggest to the smallest, unless, of course, you have the sexual chromosome, which is different, that stays there on the right, that you're going to see that in a moment. So here we have an image. This here is of an organism that have 23 pairs of chromosomes of 46 chromosomes. Which organisms do you know that have 46 chromosomes or 23 pairs? Yes, if you are paying attention, human beings. This may be human beings or maybe something else. So what do we have here? We have pairs of chromosomes, so one after the other. As you can see over here, for example, these are homologous because they have the same length and they have similar band patterns. So the homologous chromosomes, they are actually kind of a copy of each other with small variations because one are coming from mother, one are coming from father. Mother gives you blue eyes, father doesn't give you the level for the blue eyes. So they may be slightly different, but the band patterns and the size is going to be absolutely the same. Unless, of course, you're talking about the sexual chromosomes. By the way, what do we see here? What do we have here? This person, what is it? Here. We have 2 X chromosomes, and I bet you already know this. This is going to be a female. What about this one? Is there something here? Pause the video now and take a look to see if you find out something here, which characteristics, what this karyotyping can tell you about this person. Okay, now that you pause it, you probably have realized that this here is a female, have 2 X chromosomes, no Y over there, and there's something else. This person have 3 chromosomes here on the 21. So this person has what you call the trisomy of the 21, has 3 pairs of 21, which is known as Down syndrome. So Down syndrome is caused by an issue in the cell division that the person receives 3 chromosomes number 21, and having 3 changes some characteristics of the person and causing what you have here, Down syndrome. These issues with chromosomes, they can happen. They are not very uncommon in human beings, but they tend to increase with the age of the mother, especially after the age of 30. Okay? Until the age of 30, there's a very, 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 very little increase on the incidence of chromosomal anomalies, but after the age of 30, it starts increasing drastically. Okay? It can go from very little, almost 0%, and it goes to above 3% of the case that are going to be having a chromosomal abnormality. So the older the mother, bigger the risk of having chromosomal abnormality like Down syndrome. And we find those chromosomal abnormalities doing a karyotyping. Also, like we find the gender. There are techniques for you to know if your baby has chromosomal abnormalities. In Brazil, there's not much of a point for this, because if you find out that your baby has a chromosomal anomaly, there's absolutely nothing that you can do. The law doesn't allow for you to terminate a pregnancy because of chromosomal abnormality. But there are some techniques. One of them is called amniocentesis, where you use a needle to get liquid from the amniotic fluid, and the amniotic fluid they are going to be having baby cells. So you can analyze that to see if the children has a chromosomal anomaly. Another technique is a chorionic villus sampling that's done through the vagina, entering the uterus and getting a piece of the chorionic villus. So basically it's a piece of the placenta, which is children's cells as well, and you can have a karyotyping done with that. Okay, both tests, they lead to a risk of miscarriage, so it's not very worth doing it, because you can have a miscarriage because you're doing this kind of stuff, and there are some dates that you're going to be doing. And as I told you, in Brazil, there's absolutely nothing that you could be doing if you have this information. So, yeah, okay. So I hope you had fun. I hope you understood what chromosomes are and how they're going to be playing, because next class we're going to be using this to show cell division in preparation to understanding genetics. Bye-bye, and I see you in another class.